All right, thank you. So today we're going to talk about a bit of spatiotemporal modeling of cancer growth and validation with 3D cell culture data and a weird phenomenon of sedimentation that we observed in the 3D cultures. But let's start first with some introduction about 3D cell culture experiments. We are interested about them because they produce tumor growth behaviors that are similar to the in vivo ones. And at the same time, data acquisition is much more flexible than the in vivo experiments. Also, if you allow me, it allows us to model tumor growth in what we call mesoscopic scale, which we can define as a small dimensional spaces, also a small number of cells from 1,000 to tens of thousands. And another thing, we have some level of sparsity and continuity. And all of them makes both continuum and discrete models to work well in this scale. However, when it comes to calibration and validation, both of them have strengths and weaknesses. For example, the continuum models can have fast calibration, but they cannot provide more detailed quantitative information about the morphologies of the tumors. And on the other side, the discrete models are more expensive, can be slower in terms of calibration, but they can provide more details in terms of the spatial distributions of the cells. So our goal here is to take the best of both worlds and use a hybrid discrete continuum model in order to calibrate and validate it with 3D cell culture data. So we came up with this framework that you see here that takes advantage of the capabilities of both of a hybrid model in order to calibrate it and validate it with 3D cell culture data. So it consists of three parts: the experiment, the modeling, and the analysis and validation part. In the experimental part, we do 3D cell culture experiment. We segment each cell individually, and then we convert the 3D cell coordinates into density profile. And we use these profiles to calibrate our the continuum model using Bayesian inference. Then we take the calibrated model, we hybridize it, and we convert it into a cellular automaton, and we perform spatial analysis, and we compare the in silico 3D coordinates that we see here with the in vitro ones. But as you can see here, I have a question mark on the macroscopic model, and I can tell you that before we start our 3D culture experiment, we hypothesized that a reaction diffusion model would be sufficient for our work. However, biology keeps surprising us, because as you can see here, we started with this geometry where we have matrigel mixed with triple negative breast cancer cells. At day 0, they are uniformly distributed in the matrigel, but at day 14, they end up at the bottom of the space. So we set out to examine this kind of behavior based on several hypotheses, whether we have passive or active migration. On the passive part, we can have gravity or ECM compression, while on the active migration, we can have individual or collective migration, where the cells utilize their migration machinery in order to move in the space. So the first thing that we did is that we wanted to examine the effect of passive migration, let's say the easy one. So we did this by knocking out any possible effect of active migration. We did this by introducing the drug paclitaxel in our 3D cultures. Paclitaxel fixes the microtubule of the cells, so it freezes them in the space. We introduce it on day 5, and as you can see in these histograms of the number of cells across the Z dimension, initially they are similarly distributed, but at day 14, the control, the untreated samples have gone to the bottom, but the treated, paclitaxel-treated samples maintain elevated positions. And you can see also on the right panel, the 3D coordinates of these samples. And as a result, gravity or ECM compression, the passive mechanisms of this hypothesis, are not sufficient to drag the cells at the bottom of the space. So we have ruled out the possibility of passive migration. And now we performed another experiment, similar conditions between control and paclitaxel treatment, in order to find what type of active migration we have in our experiments. So the differential gene expression analysis here gave no signs of EMT, but we had more clear patterns of collective migration regulated by MAPK, TGF-beta, adherence junctions pathways. And what is interesting with this type of migration is that we have intercellular signaling. So based on this, we constructed a hypothesis that will lead us to the model design at the end. So the hypothesis starts from the fact that the triple negative breast cancer cells are adherent, so they want to be attached to somewhere in order to survive. So as we had this uniform distribution of the cells at day 0, we hypothesized that the ones that were closer to the glass bottom of the plate, they sensed the glass and they attached to it, and then they emit some signals like chemoattractants in the 3D space. The cells above them sense these attractants and emit in a similar manner, like an avalanche effect. And as a result, a chemotactic gradient is created from the bottom to the top, and the cells navigate based on this gradient. So we formulated this hypothesis using an advection reaction diffusion equation, where U here is the cell density, cancer cell density. It depends on diffusion terms, logistic, the treatment, the cell death due to the treatment, and the advection terms that drags the cells at the bottom. That depends on the gradient of the signals F, and the signals F diffuse in the space only with the diffusion term. So we move on to the calibration of this model, and we perform Bayesian inference with Monte Carlo techniques. And as you can see, here are the results on the right panel of the best parameters that we obtained, where we have a slight agreement between experiments and simulations with green and blue respectively. At day 2, control conditions, day 14, they go at the bottom, and as we increase the treatment dose, we see that they maintain elevated positions, and also the behavior of the posterior distributions of our parameters are consistent with what we would expect it by the effect of the treatment. For example, the diffusivity du on the top left and the chi, the advection constant, are consistent. They inhibit this kind of migration. Next up is the hybridization of the model from continuum to discrete. We perform this using techniques that have already been done by Sandy and some modifications later, Franzen et al. And you can see the result of the calibrated model now in its discrete versions, where we here see the 3D cell coordinates between experiments and the hybridized model with respect to the dose at the last day of our experiment. And next up is the spatial analysis, where we can compare the patterns from in silico and the in vitro 3D coordinates. Briefly here, we use the Ripley's K function. I will not go into details, but whatever you see above this 0 line denotes clustered patterns, below this line denotes dispersion, and the 0 line denotes plus some uncertainty, denotes the theoretical uniform random distribution. Okay, the different colors here are the different conditions from control, which is the blue one, to the yellow, which is the highest dose that we had in the experiment. And we can see some similarity between experiments and simulations, but more importantly, it allowed us to relate these produced patterns with respect to the mechanisms of our model. For example, here, we took the area under this K function. In the experimental data, we had this increase in this clustering index with respect to the increase of the dose. However, in the simulations, when we had a uniform cell death probability, we had the opposite behavior. So by taking a distance-dependent cell death probability, we were able to reproduce in some way the experimental observation. And also when it comes to migration, if we tweak a bit the diffusion and the advection values, we get different clustering patterns. Okay, so the conclusions of this part is that this experimental computational framework allowed us to perform fast calibration on the continuum scale. and a more spatially focused validation on the discrete scale and relate the mechanism between the mechanisms of the morphology and spatial patterns. Sorry. So, however, we still have one hypothesis that, okay, fits well to the data, but we need to convince ourselves whether is a concrete hypothesis or we are just doing it because it's exciting. So we talked about signals stemmed from the bottom of the space. So now we questioned what would happen if we introduce signaling all over the space, coming from all directions. And here we did this by doing triple negative breast cancer cells and fibroblast co-cultures, where here we introduced the fibroblasts in the adjacent space of the matrigel, and you can see they form these sea urchin patterns that hug the matrigel, basically. The fibroblasts mixed with the cancer cells, they invaded the matrigel, and as you can see on the last box plot, the co-cultures, the cancer cells maintain elevated positions with respect to the control experiments that we saw before. So the migration was reduced or inhibited in the presence of fibroblasts. Another thing that we talked about is about the adhesiveness, the adhesion of the glass. Glass is a highly adhesive surface and promotes cell attachment. Now we examined what would happen if we hide this property by putting agarose in our experiment. So we did a bit of a Boy Scouting experiment here. There is no protocol for this. We put agarose, we created some bubbles. We waited for these bubbles to solidify. We popped the upper part, and then we placed the matrigel in this pocket that we created in the bubbles. And as you can see, by hiding the adhesiveness of the glass, the cells also stopped migrating towards the bottom of the space. So the conclusions here is that, okay, we may have some pretty good evidence about this self-generated chemotactic gradient that dragged the cells towards the bottom of the space, because the model, okay, fitted well to the data. We saw that passive mechanisms were not sufficient to drag the cells at the bottom, and the co-cultures and the agarose experiment also gave us some insights about the role of the signaling and the adhesiveness of the glass. And having said that, I would like to conclude with my acknowledgements to my collaborators who were kind enough to train me to do the experiments, but they were not kind enough to do the experiments on their own. My big lab and my funding agencies. And this is my self-promotion page. So thank you very much.
Could you speculate on the nature of that chemotaxis agent produced by plastic surface that stimulates migration of the cells, and also how agarose, that is quite permeable to small molecules, stops this signal, and whether your cells were migrating sideways to the side of the wells, which also have the plastics?
Yeah, that's a good question. And honestly, I wanted to check everything that you said, but I didn't have the time to perform these experiments. So yes, testing different materials and different types of adhesive properties from the bottom would be something very interesting. Also, another thing that we were thinking about is creating a sandwich model of glass on the bottom, glass on top, and see what would happen. But this would be a bit difficult to manufacture it. So we haven't done it yet, but it's one of our goals. Thank you.
Very interesting. And pretty much following up on the last question, chemotaxis, response to a chemical gradient, is one way to explain the data, but the other way is that there's just random movement with an adherent surface. And so over time, cells just accumulate through random movement at the surface.
Definitely, yeah.
So, and your experiment with two glass on either side actually would allow you to look at it because it doesn't really require a chemotactic signal for that result to be obtained. And agarose, your experimental design with the Matrigel and the agarose, fantastic idea, really great idea. But agarose actually requires cells to be able to break down the agarose in order to migrate through it, right? That's the classic, in the classic bench assay for transformation.
I see.
So it may be that you just have cells that are not able to penetrate the agarose, which you could determine by just layering the cells on top of the agarose and seeing whether they penetrate in.
Two things here. So first of all, if we had only diffusion, the simulation would show us that the cells would also gather at the top and not only at the bottom.
Except that there's an adherent surface at the bottom that they prefer, so that the result you have here is consistent with differential adherence.
Actually, in our simulations, we took into account this type of adherence in our cellular automaton, and we saw that indeed we would have cells remaining at the top. Not any of them, every one of them would go at the bottom of the space. But yeah, that's a good point. Most of them would go at the bottom, but there would be some of them elevated. The other thing about agarose is that we didn't plan for the cells to penetrate it, but we wanted to see if they are going to still accumulate it at the bottom of this agarose.
So thanks for that clarification. Really good.
So can you tell me a little bit more about the experimental setup? How thick is the gel that you make?
Yeah.
And what is it Matrigel or?
It's Matrigel.
So what's the concentration?
Sorry. Okay, I have this small picture here. So the Matrigel has this semi-ellipsoid
what's
geometry.
The size and what's the dimension?
The dimensions is 2.5 times 2.5 the planar dimension times 0.9 millimeters.
Okay, so about 200 microns.
Yes.
Right?
Sorry, 900
microns.
Yes.
Okay, so because very likely, so there are experiments where what they do is they make these kind of surfaces, put the cells, and instead of, so they'll start off with a soft substrate, and then they'll cross-link it, so the substrate becomes stiffer, and you will still start seeing the cells migrate. And so that's more durotactic rather than chemotactic.
Definitely, durotaxis is something that we also speculate. We are not excluding anything regarding durotaxis, but in here we wanted to see the effect of this kind of chemotaxis. But of course, there is also might be some type of durotaxis going towards.
And I think the way to find that out is your velocity term and the spatial distribution on it, right? Because cells can sense up to a certain distance, both chemical signals and mechanical signals. So I think how that velocity term changes spatially.
Yeah, exactly. Regarding durotaxis, I have a comment here is that the contact guidance that is stimulated by durotaxis, the distance is around 20 micrometers long. So the cells can move with respect to aligned fibers around 20 micrometers.
Haptotaxis.
That's okay.
So contact guidance is a separate
one.
One,
different type. Okay.
One,
sorry.
Yeah, we can chat.
Yeah, thank you.